Well, good afternoon. This is a glioblastoma. Probably not all of you are familiar with this kind of disease. You can easily note that this is different than a healthy brain and also different from a stroke. You can see this is a T1 post-contrast weighted MRI. You can see this ring enhancing a necrotic core and a surrounding edema, and the mass effect. You can see that this ventricle is more, the volume of this ventricle is smaller. So, glioblastoma is a malignant gliomas. The prognosis is very poor, with a median overall survival of less than a year and a half. There are some main prognostic factors that are commonly used in clinical practice and in clinical trial, which are age, performance status, extension, surgery, resection, EDH1 mutation, and GMT promoter methylation. The treatment is a combination of surgical resection, chemotherapy, and radiation. But you can easily imagine that this kind of mass will have an important impact on the structure of the brain, in particular the white matter tracts. But currently, to date, there are no prognostic factors based on the impact that glioblastoma has on white matter tract. This is unsure that more than 80 here, this is the most important pathologist of the last century working on glioblastoma. And in more than 80 years ago, he pointed out the fact that probably the most important thing that was to study in this disease was the pattern of spreading and growth this disease. So how this disease was spreading. But probably in the last 50 years, oncology research focusing other aspects, not in the pattern of spreading of the disease. And but the pattern of spreading is very interesting for us, that we are neuroscientists, and new results discovered that the spreading of glioblastoma is connected, is linked with the organization of the structural organization of the brain. For instance, malignant cells they spread climbing along white matter tract, along exons and along white matter tract. Can you please start the video? Thank you. Thanks. You can see here in vitro how the malignant cell climb and spread along exons, white matter tract. So you can easily figure out how the structural organization of the brain is relevant for the pattern of spreading of the disease. And in the last very few years, some studies found how there is a mutual interaction between gliomas and neurons and the brain. In particular, there are secretion of excitatory molecules as glutamate from the tumor and cause an increase of activity of the neurons, and activated neurons promote the growth and the spreading of the glioblastoma. So there is this neuron activity dependent progression of the disease or the tumor. In particular, it is very interesting. There are the creation of neuron to glioma active synapses. Well, we hypothesize that what happens in the macro scale, so the creation of this neuron to glioma synapses, the integration of the glioma cell in local network, will have an effect also in a large scale, so at whole brain network. And so that we could measure this effect with the whole brain connectivity approach. This is not a very new concept. This is Galeno, an ancient Roman physician. He postulated that the pathology in one part of the nervous system could spread in other region. He told about animal spirit that flow through interconnecting neural pathways, but it is not much different than the connectomics approach on brain disorder. In agreement with this very new, very novel way to see the disease. The connection topology of the brain shapes the onset, expression, and the progression of brain disease. So there is an interaction between the structural and functional connectivity of the brain and the disease, the pathogenic mechanism of the disease. But how glioma cell alter functional connectivity? There are different processes. One is the fact that tumor can damage neuronal connection, and this will result in a decrease of functional connectivity. Then the possibility that there is a disruption in neurovascular unit, so the both signal is lost, and so again there is a reduction in functional connectivity caused by the tumor, but also there is an active connectivity between glioblastoma and glioma and the surrounding brain. So there are different kind of mechanisms that potentially can result in a different effect, an increase or a decrease on connectivity in the surrounding tissue, the surrounding brain tumor. So here, now I will show you the result of some study, very recent, because this topic on the impact of glioblastoma and brain, the link between glioblastoma and brain connectivity and the impact of overall survival is a very novel topic. So the first interesting result is you can see here in green that within brain tumor there is actually functional connectivity. So very different than stroke, it is just necrosis. There is active neuron activity within a brain tumor. And it is interesting that there is the intratumoral connectivity strength is a prognostic marker of overall survival. In particular, the higher is the connectivity within the tumor and the longer the overall survival. There is another work showing that homotopy functional connectivity from the tumor is a predictor of overall survival. In this paper, this is a group, an Italian group, they found that they divided different clusters of connectivity from the tumor, or seed-based connectivity from the tumor to other regions of the brain. These three clusters are right frontal, left frontal, right occipitotemporal, and so on, but these clusters are significant. You can see these clusters show a significant correlation with overall survival, but it is interesting that there are two clusters with in which the relationship is in a direction, and a cluster in the opposite direction. So it's not really clear what happens in this mutual interaction between brain connectivity and tumor activity. But what is interesting is that the functional connectivity measured from the tumor is a stronger predictor of overall survival, stronger than all the other prognostic factors are commonly used in clinical practice and in clinical trial. And the same for the structural connectivity, the impact that glioblastoma has on structural connectivity. In this recent paper published in Brain by the this Cambridge group, they found that they computed the impact that the tumor had on structural connectome at pre-surgical, before the surgery, and they found that the larger was the impact on the structural connectome, the worse was the survival. And this prognostic factor was significant also in different kind of model. It was, the significance was comparable or than the other prognostic factors. And interesting, the distance of the structural connectome alteration was correlated with the distance of the recurrence of the tumor. Yeah, now I will show some slide on a study that we performed here in Padova. A couple of years ago, with Lorenzo Pini and Professor Corbetta, we started thinking about the problem, considering that the brain is not the same. The white matter fibers are not the same across the brain, because there are regions where the density of fiber is low and region where the density of fiber is high. So there are regions that are more peripheral, less connected in the brain system, and regions that are more connected where the density of the fiber is higher. So same wise, there are glioblastoma that are located in region of high density of fiber, and glioblastoma located in region which are less connected with the rest of the brain. So we hypothesize that the glioblastoma located in region of high connectivity were associated with a worse prognosis, because the more possibility of spreading, more exons, more white matter tracks to spread. We computed this index that we call track density index. We use an atlas from the Human Connectome project with hundreds of tractography, and then we computed for each single voxel an average of the number of white matter tracks passing from each of these voxels in order to obtain maps in which for each voxel we have the number, this average number of white matter tracks passing through. And then we just, for each of our patients, overimpose the ROI, the mask, the two-dimensional mask of our tumor, the tumor of our patient, and computed the track density index in each voxel of this. We just sum the value of track density index for each voxel on the mask. So we obtain a number for each tumor that indicate that that tumor was located in a region of high density or in a region of low density of white matter fiber. So we performed this study in two completely independent cohorts. First we assess the relationship between track density index and overall survival, and then we test the ability of prediction of overall survival from track density index. You see here in red, the red dots represent the correlation between overall survival and track density index in the two data sets. And you can see that there is a significant correlation. In particular, the higher was the track density index, the lower, the shorter was the overall survival. Then in orange, here in the right, you see in orange the stability of the correlation. This is the correlation value 0.4, the correlation value between track density index and overall survival. You see that the line in orange represents the track density index. It is stable, independent from the length of the survival of the patient. You consider the other prognostic factors that I repeat are commonly used in clinical trial, in clinical, in everyday clinics. The other prognostic factors are not stable and reliable. When the survival is longer, the correlation dropped to zero. So we found that this track density index strongly correlates with overall survival in both data set, and this relationship outperformed the association between the classical prognostic factor. We also performed some Kaplan-Meier curve dividing the patient, grouping the patient between patients with high track density index and lower track density index. The survival was different, significantly different, and also there are some multivariate model where when we added the track density index to the other prognostic factor, the prediction was significantly higher. And then we test out of sample the accuracy, and it was and also higher. Then some additional analysis to exclude the effect of the volume of the tumor, the location of the tumor, and the surgery extension. So we confirmed that there was a significant relationship between the glioblastoma impact on the structural organization and the overall survival, just confirming that the white matter may represent the structural pathways from glioblastoma spreading. So once again, there is a strict relationship between the intrinsic organization of the brain as a structural connectome and the effect of glioblastoma. So it is interesting that this index can be used in clinical practice because you just need the T1 images. You just need a segmentation, so you can you could you can obtain this index from clinical imaging. You can obtain this index at pre-surgical level, so the surgeon can potentially tailor the surgical strategy according to the prognosis of the patient. I think we will skip this. I just tell you that we try to estimate the survival using an indirect approach, functional indirect approach. We told a lot yesterday about this, but we didn't find any correlation with the indirect functional connectivity approach. So it is just to summarize that there is a strong relationship, a strong predictor from structural connectivity, functional connectivity, and the tract density index, that is an indirect method of structural connectivity of overall survival in glioblastoma. Of course, once you found that there is a strict relationship between connectivity and prognosis of glioblastoma, you can open new opportunity for treatment, because you can interfere with this connection in order to modulate, suppress synaptic signaling in the neurons surrounding the glioblastoma in order to slow down the migration rate of malignant cells. Moreover, you can tailor the radiotherapy, the radiation treatment. Currently, radiotherapists just irradiate, just do the radiation in the radiation field is measured just in terms of they radiate 2 centimeters in the surrounding tissue, just measuring brain in centimeters, but brain is different from 1 millimeter to the other, so we need to tailor the radiation treatment. A tract density index may provide a way to improve the tailoring of this treatment. And more in general, this is the last slide, there is a sort of paradigm shift in the way we look to glioma and glioblastoma, because in the last 50 years, the research focused on glioblastoma itself, the immunology of glioblastoma, the genetics, the molecular aspect of glioblastoma, but completely didn't look at the fact that glioblastoma is located in an organ that is a special organ, the most interesting organ of our body, it is the brain. And the brain is not just a host, but actively interacts with the tumor, and the interaction is mediated by the connectivity. And so if we look to this disease from a connectivity point of view, probably we can open new opportunity for the treatment. And there is a new field, it is very new, it is very promising, that is called cancer neuroscience, that put together all the concepts that I tried to explain in this presentation. So thank you for listening to me, and I also like to thank all the Professor Maurizio Corbetta, Lorenzo Pini, the team of Alessandro Bertoldo, and all the other people you can see in this slide. Thank you.